Hey, my name is Lukas, and I'm glad that you're here to watch this little introduction video about TANGLE, which is our transcriptomic-guided slide representation learning algorithm, which can be applied in tasks for computational pathology. So let me walk you through the design of TANGLE in particular. So where we start from is from histology slides, and these histology slides are usually gigapixel-sized images. What does that mean? So we can't basically throw them into a vision transformer and process them since they are too big. So the goal of TANGLE is to process them in a hierarchical way to infer a slide embedding, which it should be a numerical representation of the histology slide. And this should be ideally done in a self-supervised manner, since we don't want to require human annotations as we have a lot of data. And once we can infer the slide embedding, we can do a bunch of cool stuff with it, since it enables us applications with minimal data usage. For instance, we can think about few-shot learning on slide classification, whether we have, for instance, a cancerous tissue sample or a healthy one. Or we can also think about retrieval tasks where we can query from a huge database and find similar cases. Exactly, and as I said, as we are processing in a hierarchical kind of way, we don't want to lose information on the way. So we want to, in this slide embedding, we want to include informations from the region as well as from the patch levels. And these patches are in our case 224 times 224 pixels. So let me give you a better view of what's going on during the training process of TANGLE. So let's start from the histology slides. And since they are gigapixel-sized images, we want to subdivide them into smaller patches. And we can tokenize these patches and feed them to a vision transformer. And this vision transformer basically serves as our feature extractor. And we can exchange it since it's frozen, it's not trained during TANGLE, and we pre-trained our own model. But this is not really what we want to do, since we now have a pooling of the patch embeddings, but they are not really contextualized. So what we want to do in the next step is to assign weights or importance to the patches. And that we do with an ABMLL structure, which is basically a weighted summarization of the pooled patch embeddings. And we can, or the model during the training process learns to assign the importance of the patches. And this can be used as a summarization, as I said, and we output, therefore, our slide embedding. So we end up with a single numerical vector, which includes information from all the whole slide image. But we are not there yet. So we need somehow a supervision in our training. And that's where we use our bulk RNA sequence data as a training signal. So for each slide, there's a vector where we have gene expressions, and we can normalize them and subselect a few important genes. And we can also like drop them into an MLP to get the same dimensionality as the slide representation from the upper branch. And so we end up having two numerical vectors, and they we can align pretty easy during a self-supervised learning, contrastive learning step. And that's what we do, and that's how we train TANGLE in the end. During training, TANGLE uses the histology slides as input, as well as the bulk RNA sequence data as a supervision signal. But the cool thing about TANGLE is we don't need the bulk RNA sequence data during inference anymore. So we can just throw the histology slide in, and what it would output would be the slide representation, and if you want, also the patch features. And this can be used now to infer and visualize the important regions which the model identifies, um, as similar to the bulk RNA's transcriptome. And we can see along the way when we zoom into regions and also on the patch levels what's actually going on there. And in this case, we are being able to identify hepatocellular hypertrophy and fatty change in the patches as well as in bigger regions. But that's not everything we can do with TANGLE during inference time. So imagine the case, as I said, we don't need the bulk RNA transcriptome transcriptomic data during inference, but if we have them available, we can also throw them into TANGLE. And what it would output would be a representation of the bulk RNA sequence data. And once we have this, we can backtrack the gradients and integrate them over the inference way. And that gives us an estimate of which genes are important during inference time. And that makes our model a bit more interpretable of what's going on from the medical side in our whole slide image. So this was a brief overview of what's going on in TANGLE. And to get a deeper understanding, I also linked the paper and the code base, so feel free to check it out.